Generally true, you prefer PDF over PowerPoint and to download them. So can you convert PDF to PowerPoint? So a couple of just administrative items. So one, good news is today's lecture is pretty simple. So I think you'll find this to be easy to follow. So it should be a little bit of a breather. Two, next week, next Tuesday, I was asked to go to Bethesda, where the National Institute of Health is located, because they're having a very big planning session for spinal cord injury research, and they wanted me to help participate in some of the planning. So unfortunately, I will not be here next Tuesday. But the good news with that is that an even better teacher, Lydia Debussy, who should be over here, is going to be speaking to you. The plan was originally to talk about Williams syndrome and Down syndrome because they lead to intellectual deficits and they have some interesting symptoms. So we're going to change things up a little bit, and Lydia is going to talk to you about peripheral neuropathies. So it's not related to Williams syndrome or Down syndrome in any way, but really important nevertheless. But she's going to talk about peripheral neuropathies and also what happens with regard to nerve injuries and the function of some of the specialized cells that ensheath neurons and support neurons in the periphery. This forms the basis for some of her own research. Lydia is a postdoctoral fellow in my own laboratory, and so she's going to have enormous insight. So you can you'll have a hard time responding to this question. The midterm is coming up. Next week, I don't believe we have any papers for discussion, and so I think all of the study sections will be centered around preparation for the midterm. Is that generally the case? Okay. Good. And then I think we're getting up permission to podcast some of the study sections, so you cannot attend one of the meetings to be able to listen to the discussion. Yeah. No, I need to get clarification from the TAs myself. I believe the discussion sessions will also be held and will help in preparation. But maybe what the TAs have done is set up a special preparatory section.
So on Monday the 11th we have a discussion section on Monday the 11th we're going to have discussion sessions. We're going to discuss the plan paper. The next week we will not have any discussion sections, and that's because that's the holiday week of the President's Day weekend, right, on the 18th. So the week of the 18th, when we actually have the midterm, we're not going to have a discussion section. But we will be doing review during the prior paper section on the 11th, and we're also going to have a review session on the 16th, which is Saturday, and that's what we're trying to talk about. If you have any questions, please do.
Do you think you could write that out and post it so that people know exactly what to be expected? Okay. Is that sufficiently shaky to have it? Okay, good. So today, okay, reinstalled. Okay. Today we're going to talk about prions. I know that many of you are aware of prions. In fact, we even used the word a couple of times in the context of ALS. But it's such an important concept in terms of neurodegeneration. It's the kind of thing that you're going to see coming up over and over again. We're going to talk about a fairly specialized form of a prion disease named after two physicians, Creutzfeldt and Jacob, or Jakob. I think the guy was German, so I'm not sure how he says his last name. Creutzfeldt-Jakob disease. And we're going to talk about the fact that this type of neurodegeneration does have the radar up for epidemiologists because there is some evidence that this type of neurodegenerative disease can be transmitted from one person to another. In other words, there's potential for infection and potential for epidemic. And so there's a lot of anxiety around that. Because these diseases tend to form very slowly, it's also unclear whether an exposure event in young life might not predispose you to something really bad 20 years later, and that's also a very dangerous scenario. So let's dive in here. This is a section of brain tissue that's just been stained. If you look at the right side, these are just sort of normal neurons that you can see showing up. And if you compare that to the left, which is a sample of brain tissue that has the equivalent of Creutzfeldt-Jakob's disease, that's generically referred to as spongiform encephalopathy, or encephalitis, sorry. It's also known as encephalopathy. The first thing that you'll probably notice is that the one on the left is not quite as dark, but that's not the result here. What you should be really comparing are the holes in the tissue. Now, if you're a histologist and you're bad at cutting tissue sections, you might actually create those kind of holes accidentally, but this was not accidentally created. There are big vacuoles or holes in the tissue, making it actually somewhat spongy. And this is one of the hallmarks of this type of disease, is that you end up with these big vacuoles or open spaces in the brain. And this is caused by a problem with protein misfolding. And this is a theme that turns out to be incredibly important for the normal function and maintenance of our nervous system, our nervous system is remarkably sensitive to having misfolded proteins. How many of you are aware that we, our cells rather, have some pretty sophisticated systems for tagging proteins with things like ubiquitin when they're misfolded and they're supposed to be eliminated, and that there are whole machines, proteasomes namely, that break down proteins. But there are conditions where the proteins form very, very resistant conformations and often higher order aggregates, and those things begin to accumulate in cells, and through processes that still aren't all that well understood, begin to become toxic to the cell. So in the top group there are a number of protein or diseases that are associated with misfolded proteins. Now, we have talked about ALS, the one in red. In your slide deck, I actually forgot to include that one and added it last night, so add it to your notes. We talked about it in the context of ALS and superoxide dismutase and the misfolding of SOD resulting in the degeneration of motor neurons. But there are other examples of it. In football, from the high number of impacts that some of the players encounter, there are diseases like chronic traumatic encephalopathy, CTE, which is also probably not transmissible in the classic infectious way, but is also based on a problem with protein misfolding in the brain leading to neurological neuron loss. Some of these things will be touched on in additional lectures, but what we're going to really focus on today is the next category, the one below, and that's the transmissible forms of these encephalopathies, and namely the one I just showed you the slide of the spongiform encephalopathy. And these are encephalopathies that are seen in many different species. So in humans, it's referred to as Creutzfeldt-Jakob disease, CJD. But in cows, it's known as bovine spongiform encephalopathy, BSE, or typically as mad cow disease. Now, many of you are pretty young and may not even remember in the United Kingdom when there were nearly 200,000 cattle that had to be destroyed because they were displaying symptoms of mad cow disease. We're going to talk a little bit more about it. It was due to bad food practices and husbandry practices. But mad cow was something that was pretty prevalent, especially in some countries, on the order of 20 or so years ago. So BSE. Scrapie is a disease that is seen in sheep, and it has been around for a very long time in sheep. And the reason it is called scrapie is that, unfortunately, one of the side effects of this disease in sheep is that they develop this terrible itching sensation, and they scratch so much that they actually create big patches of open skin and open wounds. All of these diseases are associated with neurological deficits that are manifest as either problems with walking, so ataxias. There's usually muscle wasting, probably indirectly related to the inability of the animals to move very well, but there may be some direct effects on muscle as well. And there are some pretty severe behavioral changes in the animals, which is where the mad cow business comes from. In humans, the behavioral things are a little bit more obvious. So one of the early signs is often a change in the patient's personality, but it's usually rapidly followed by indications of ataxia and dementia in humans. So sheep, cows, humans are three examples of spongiform encephalopathy, but in fact, you can replicate the disease in mice, and it's observed in mink populations, deer populations. So probably most mammals are, to a degree, susceptible to this kind of transmissible spongiform encephalopathy. So prions, as I'm sure most of you know, are kind of well-known infectious agents because they lack nucleic acid. And we'll go over a little bit of the kind of histrionics and the research that was associated with that concept. But it was a pretty remarkable concept in its day that you could have an infectious agent that had neither RNA or DNA in it. But the prion name itself is derived from this notion that you could have a proteinaceous infectious particle. So that was sort of coined as the term prion. It leads to the neurodegeneration and the big vascular flows that you saw in histological sections. Another feature that is different about this infectious disease compared to many types of infectious diseases is that it's slow. And so a classic form of an infectious disease, a virus, for example, would be over the course of days and weeks, there would be an infection, there would be symptoms, there would be an inflammation. The immune system would recognize the foreign object, and usually symptoms get better in most cases. In this case, the disease is slow, so maybe on the order of years, and there's no inflammation. For people who work on infectious agents,
that put this in a category that was so absurd, it was very hard for them to even conceive of these types of diseases as being infectious. So slow and no immune response were just completely counter to the way that people would normally think about an infectious agent. And then we've already talked about some of the histological, or you saw the section of the histological things, tissue. So you find that there are these big spongy or vacuolar holes that develop in the nervous system. In terms of the symptoms of humans, I touched on that a little bit. So ataxia, dementia, personality changes are some of the classic forms. And in humans, the disease CJD, or Creutzfeldt-Jakob disease, can appear sporadically in individuals. It's a rare disease. I should maybe start by saying that. So on the order of 1 in a million, which puts the risk factor or the frequency in the U.S. at about 300 cases per year, which makes it exceedingly rare. About 85 of them are sporadically arise, and about 15% can be traced back to a gene mutation that was inherited. We'll get into I've already gone through some of that. In terms of the historical perspective here, so in sheep, the spongiform encephalopathies described as scraping in sheep were recognized for hundreds and hundreds of years. So this has been something that's endemic to the sheep population. And in sheep, it's probably a disease that can arise sporadically at a fairly high frequency, and then is probably spread among sheep pretty efficiently. And the main route that is thought to be spread in sheep is one, either feeding them contaminated material in their food supply. And so this is a theme that will come up in the context of mad cow disease as well. But unfortunately, what happens in the meat processing business is that all of the scrap material, bone, and nervous system, and so on, is often gone to rendering works, where I guess it's all sort of cooked in the thought that high heat is going to kill anything that might be infectious, and put back into their food supply as a source of protein. And it turns out that prion diseases are pretty resistant to heat, and so the rendering process is not sufficient to inactivate this infectious agent. In the 1700s, it's unlikely that they were doing that. It wasn't big agra at that time. So in sheep, it was probably also the case that they're quite sensitive to developing it sporadically, and sheep as an animal eat their placenta after a birth, and that's one of the ways that it was thought to be spread so commonly in sheep. So in the 1920s, Creutzfeldt and Jakob were physicians, and they diagnosed the disease in humans. And then in 39, it was demonstrated that you could transmit scrapie from one sheep to another. And so this was one of the first clues that this might be an infectious disease. There was no clue that it was proteinaceous at the time. And then in the 60s, this link was made between a disease among Kuru tribes in New Guinea and Creutzfeldt-Jakob disease in other populations of humans. So let's talk a little bit about Kuru. So Kuru was a sort of is a bizarre story, let's put it that way. So in the 60s, it was a tribe of people living in the so-called Fore area, F-O-R-E, area of New Guinea, were identified. It was a very primitive tribe of individuals, and they practiced cannibalism. And it wasn't the sort of cannibalism where somebody would get hungry and decide to go over and eat their neighbor. It was the type of cannibalism that was a little more civilized, if there's such a thing, and that is, it was a ritualistic type of cannibalism, in that in order to honor the dead individual, everyone in the tribe would eat a portion of the individual. And what was recognized among this tribal population of people is that they were developing a disease that they called kuru, which is, in their language, was described as a shivering kind of disease, and that was really their way of describing the ataxia. It was pretty common in their children, and it turns out that it was pretty common in the children because of their practices in cannibalism. So the males in the tribe would tend to get the better cuts of meat, and so they would eat muscles. And the women and children would sort of be relegated to eat things like the brain. And the brain is the source of the highest concentration of the infectious prion particles, and therefore the children and women were the ones who were most often diagnosed or developed what the tribal people call kuru. And so it had the classic symptoms that you would normally associate with Creutzfeldt-Jakob disease. It had the ataxia and dementia. And it also had this sort of slow feature. It took time for the disease to develop. There wasn't an immune response. There wasn't any particular obvious kind of inflammation that was taking place. So there was a researcher named, he was called Carlton Gajduszek, and he became interested in this type of infectious agent because it was breaking these rules, as I touched on. It was breaking the rule that an infectious agent could manifest itself as a disease slowly rather than very quickly, and that it seemed to be evading the body's immune response. And consequently, there was a real debate. Is this truly an infectious agent, or are we dealing with something else? So Gajduszek was not interested in the business of proving whether this was protein or nucleic acid. He was interested in showing that we were dealing with an infectious agent here. And so he went to New Guinea, he lived with the people, he studied them, he recognized the fact that they practiced cannibalism. That was sort of an obvious potential way of transmitting an infectious agent from one person to another. The more classic way, of course, is for somebody to sneeze on you, or what is known as the oral fecal pathway for transmitting the disease. But by going and studying this particular tribe, he learned a lot about the disease itself, but he also collected samples. And so he collected autopsy material from, in this case, an 11-year-old girl who had developed and died of kuru. And he took that back to the National Institute of Health, where he was running his laboratory. So this is in the 70s. This was one of the places that could really afford to do primate research. It was a very expensive type of research. And he took samples from this girl and injected it into 2 chimpanzees. And then he waited. Okay, so in the typical business of research now, if you had to wait 2 years without a result, you'd be pretty nervous. You'd be worried that you wouldn't be promoted, you'd be worried that you'd lose your job, you'd be worried that you'd lose your grants. But this was a little bit kinder, gentler time in research, and basically waited 2 years. And over the course of the 2 years, the chimpanzees developed something like Kuru disease. And this had some very, very important implications. So one, it showed that this was a disease that could be transmitted. And two, that it was capable of crossing a species barrier. So as you know, a lot of pathogenic agents do not jump from one species to another. Now, the flu virus is an example of something that kind of evolves going from, I guess, chickens to pigs to humans. But a lot of infectious agents are not able to cross species barriers very efficiently. And then it was also atypical in the sense that even in this experimental example of him transferring this infectious agent from this girl to chimpanzees, the chimpanzees did not exhibit or display the classic signs of inflammation, despite having jumped from one species to another. So this was actually regarded as really significant research that you could have this type of infectious agent. And so he was awarded the Nobel Prize in 1976 for these really landmark studies. And over the next decade or so, Carl von Geiterschek was regarded as quite a humanitarian because he adopted over 50 children from New Guinea. But when, I guess, he got to the number 50 or so, people started wondering why so many. And they started looking at the pattern and realized that all of them were boys. And this turned out to be a sort of tragic story because in 1997, he was prosecuted for child molestation. So the guy had some serious personal problems as well. And he spent quite a period of time in jail, but he passed away in 2008. And so while he made some important contributions to science, unfortunately, this isn't the most pleasant story to have to tell. So let's talk a little bit more about mechanism and a little bit less about cannibalism and pedophilia. Scrapie. So this is a sad image of a sheep that's been that has the disease, hence the name Scrapie. That in the United States, it's sort of a 1 in a million disease, so that's where the number 300 or so comes up, since we have about 300 million people in our country, so it's a very rare disease. This is just sort of a review of some of the things that I have described. So there were these parallel lines of research that were going on with sheep and trying to transmit the disease, as well as Geiterschek's work with Kuru, showing that the disease could be transmitted, and relating this to Creutzfeldt-Jakob disease and a couple of other very related diseases that have this. These are the names: the Gerstern-Straussler disease and familial insomnia. They're basically the same thing as Creutzfeldt-Jakob disease. So in the story sort of moves into the 70s, and Stanley Prusner was a medical doctor at UCSF, and he was a young doctor. He had a patient come into his office who he diagnosed with Creutzfeldt-Jakob's disease. And I guess because he was young and energetic and had enough time on his hands, he wanted to learn more about this. And in part because it was so extraordinarily rare. And so he started reading about this disease and trying to understand it more. He knew about some some of Gajdushek's work, but he'd also heard this rumor that maybe it was a new kind of infectious agent. And at this time, this was a revolution in molecular biology. There was enormous focus on oncogenic viruses, for example, RNA viruses, and DNA viruses. And so this one seemed kind of interesting. And so he had a research laboratory at UCSF, and he became so interested in this patient and mechanisms that underlie CJD that he began to do research. And one of the first things that they wanted to do was set up a reasonable model for studying the disease. And so up to that point, if you wanted to model the disease, you'd work with sheep. And sheep are big animals, and keeping them in a city is not that straightforward. And so they worked out a new model system in which they showed that you could purify scrapie particles from sheep, and you could put them into hamsters, and the hamsters would develop something like scrapie. And so they were able to transfer it from sheep to hamsters, and hamsters were a much more convenient research model for them to be able to work with. Having these purified protein fractions and having an animal that they could sort of reliably go to and test their purification strategies was a huge step forward. So often in the purifications, if you don't know what you're purifying, it's very empirical. You run fractionation columns based on size and other properties, but you don't know which fraction the agent is going to come out in. But having a system in which they could test the hamster model was very important. And so they fractionated and began to purify scrapie, and using their bioassay, the hamster, as a way to detect which fractions had the highest concentration. And they developed very highly purified samples of the infectious agent. And that allowed them to then begin to ask some basic questions. So they really wanted to test the idea whether there is RNA or DNA in there, and could they begin to clone it and characterize it and understand what kind of infectious agent it really was. But this quickly led to the observation that the classic things that you would associate with RNA and DNA weren't really so easy to demonstrate with these preparations. So one of the things that you would classically do is to take a sample of highly purified infectious agent, of scrapie in this case, and if it has nucleic acid in it, you can hit it with a particular wavelength of ultraviolet light, and that will cross-link the nucleotide bases.